Hello everybody, so let's just discuss what celiac disease is and what is the pathogenesis of celiac disease. So what is celiac disease? Celiac disease is also known as celiac sprue, gluten-sensitive enteropathy. By the word gluten-sensitive enteropathy, we mean that there is some kind of intestinal entero word is for intestine, right? Intestinal, and pathy means pathology. There is some kind of intestinal pathology due to gluten sensitivity. Okay, your intestines are sensitive toward gluten, due to which you have pathology. Okay, your intestines are undergoing some kind of pathology due to gluten sensitivity. Okay, so what is the definition of celiac disease? It is an immune-mediated enteropathy triggered by the ingestion of gluten-containing food such as rye, barley, wheat in genetically predisposed people. So, who is at risk, or what are genetically predisposed people? So, the people who have HLA-DQ2 and HLA-DQ8 on antigen presenting cells. Okay, they are actually genetically predisposed people, and they will get gluten sensitive enteropathy. The physiology of gluten, and I will read the text with you after that. Okay, suppose this is your intestine. Okay, and you know that you have 4 layers of your intestine, right? You have mucosa, submucosa, muscularis externa, and then adventitia, right? So this is the epithelium. Okay, and mucosa is further divided into 3 layers, right? Epithelium, lamina propria, muscularis mucosae. So this is epithelial layer, and this is your lamina propria, and okay, this is your muscularis mucosa. So this whole thing over here, this thing over here is your mucosa, right? So suppose someone ingested gluten. Okay, someone took gluten in the diet, and gluten comes into your intestine. Okay, if someone takes gluten and then gluten comes in the intestine, what will happen to gluten? Gluten will be converted to gliadin. Okay, gluten will be converted to gliadin by the action of enzymes. Certain kinds of enzymes are present in the intestine, which will act on the gluten and convert the gluten into gliadin. Okay, now this gliadin is resistant to further digestion. It won't be digested, and it will stay inside the intestine. Okay, and when it is present inside the intestine and it is not further digested, what it does is, let me make it. Okay, this is your gliadin. Gliadin will apply stress on these enterocytes. Okay, it will annoy them simply. It will apply stress. So these enterocytes are undergoing stress applied by or created by your gliadin. Okay, so what happens when these enterocytes are undergoing stress? They start secreting interleukin 15. Okay, they start secreting interleukin 15. Okay, what does interleukin 15 do? Interleukin 15 does 2 things. It increases the proliferation of your CD8 positive T cells, which are classically known as cytotoxic T cells. Let me write it. Okay, cytotoxic T cells. And interleukin 15 also increases a natural killer surface marker on the surface of cytotoxic T cells, classically known as NKD2G. Right over here, NKG2D. Okay, NKG2D. So, okay, NKG2D. Okay, it's actually G. Okay, NKG2D. So, okay, let's repeat again. gluten is converted into gliadin. This gliadin is not digested further. Instead, it applies stress on the enterocytes. Enterocytes undergoing stress will start secreting interleukin 15, and interleukin 15 will increase or stimulate the proliferation of cytotoxic T cells, and these cytotoxic T cells will also express a marker NKG2D. Okay, and side by side, these enterocytes undergoing stress will start expressing a surface marker. Okay, a surface marker. Okay, what is the name of the surface marker? Let's see over here. This is MICA, MICA. Okay, so MICA. So this gliadin, by applying stress, does two things. MICA, the enterocytes start expressing MICA on their surface, and the enterocytes secrete interleukin 15, which will increase the CD8 positive T cells, and these CD8 positive T cells will express NKG2D. Now, what happens? These cytotoxic T cells, which have NKG2D, these bind to your MICA. Okay, let me explain over here. So this is your intestinal wall, and these enterocytes, suppose this enterocyte has undergone stress, okay, stress applied by your gluten, okay, actually gliadin. Gluten has been converted to gliadin, right? So this is MICA over here, and you have increased the proliferation of cytotoxic T cells, and you have also increased or stimulated the expression of this NKG2D over here. So these two bind. Okay, this cytotoxic T cell binds the binds with NKD2G with your enterocyte. Okay, what happens now? Okay, if I ask you, how does cytotoxic T cell kill any cell or any organism it has to kill? So there are two ways by which cytotoxic T cells function. Remember this is very important, and this is standard. Okay, so two functions. When cytotoxic T cells bind to a substance or a cell to which they are actually trying to attack. Okay, so they secrete two things. They secrete coenzymes and they secrete perforins. Okay, anywhere in the body, cytotoxic T cells, when they have to kill a cell, they secrete coenzymes and they secrete perforin. Even if they want to kill a virus, they will do like this. Okay, this is like if macrophages and neutrophils want to kill some antigen, they will first phagocytize it, and then they will secrete lysosomal enzymes and reactive oxygen species. Okay, so the mechanism of function of cytotoxic T cells is to secrete coenzymes and perforins. Okay, so perforins will create holes in the cell membrane. So when perforins are secreted onto this enterocytes by cytotoxic T cells, this enterocyte will have holes in the cell membrane. Okay, and when cell membrane is damaged, you know that the cell is no longer able to live. Okay, and coenzymes will increase the activation of caspases. Okay, in the mitochondria, and these caspases are going to increase apoptosis. Okay, and eventually this enterocyte will die. Okay, so this is very important process in celiac disease pathogenesis. Okay, repeat with me. You have a cytotoxic T cell which is binding an enterocyte, and then this cytotoxic T cell will secrete coenzymes and perforins. Coenzymes will increase the activation of caspases, which will lead to apoptosis, and with perforins you are creating holes in the plasma membrane of this enterocyte, and obviously there will be instability, and this enterocyte will eventually die. Now what will happen to this intestinal wall? It was initially like this, and now it is like this. There's no enterocyte over here. So what happens, you have gliadin in your intestinal lumen, and now this gliadin will go into lamina propria. This is very, very important, and you should have the strong concept of this. Okay, so now what happens when you have something like this and gliadin goes inside the lamina propria? Inside the lamina propria, you have an enzyme to convert gliadin to deamidated gliadin. Okay, now gliadin is converted to deamidated gliadin. Okay, we are going to read the text together so everything will be clear again. Okay, so the enzyme that converts gliadin to deamidated gliadin is known as tissue transglutaminase. Tissue transglutaminase. This is very, very important. This is high yield. Okay, this is very, very, very important. This enzyme. Remember its name. Okay. So you have tissue transglutaminase enzyme in the lamina propria, which converts your gliadin into deamidated gliadin. Now, this deamidated gliadin is recognized by certain kinds of antigen-presenting cells, which have HLA-DQ2 or DQ8. Okay, DQ2 or 8. With HLA-DQ2 or DQ8, these antigen-presenting cells recognize this deamidated gliadin. Okay, this is very important. Now, what these antigen presenting cells do, they present this deamidated gliadin with their HLA-DQ2 and HLA-DQ8 to helper T cells. Okay, to helper T cells. And what will happen now? These helper T cells will stimulate your plasma cells. They will stimulate your B lymphocytes by secreting interleukin-2, and these will this interleukin-2 will convert your B lymphocytes into plasma cells. Okay, and plasma cells will make antibodies against your tissue transglutaminase or your deamidated gliadin. This is very important. Okay, so let's just read the text together. Okay, so the person ingested gluten. Gluten is digested to gliadin, which is resistant to further digestion. So it stays in the intestine. It applies stress on enterocytes, and these enterocytes, which are undergoing stress, will secrete interleukin-15. Interleukin-15 does two things. It stimulates the proliferation of CD8 positive T cells, and it increases the expression of NKG2D, a natural killer cell surface marker on the surface of CD8 positive T cells. And enterocytes undergoing stress express MIC-A. Okay, now MIC-A and NKG2D binds together, and CD8 positive T cells secrete granzymes and perforins on that enterocyte, which will obviously damage your enterocyte. And by damaging enterocyte, you are actually increasing the permeability of your intestinal wall to gliadin. Okay, this is very important. You actually increase permeability of intestinal wall to gliadin. Now, gliadin is able to go into lamina propria. Initially, your enterocytes were not allowing gliadin to go in the lamina propria. Now there is no enterocyte, and now gliadin is able to go into lamina propria, where it is converted into deamidated gliadin by tissue transglutaminase enzyme, TTG. Okay. Now, deamidated gliadin binds with HLA-DQ2 or HLA-DQ8 on antigen-presenting cells. So, HLA-DQ2 or 8 is not present in everybody. So, the people who have this on their antigen-presenting cells are susceptible to get celiac disease. Okay, now this deamidated gliadin is presented to CD8 positive T cells, and antibody is formed against deamidated gliadin and tissue transglutaminase, and this thing will cause further destruction to your enterocytes, to your intestine. Okay, so very important. So you know that on the enterocytes you have villi like this. Okay, and there are like so many villi in your enterocytes, and you have so many um, enterocytes in your intestine. Okay, so when gliadin, obviously gliadin is not going to attack one or two enterocytes, it's going to cause massive loss of enterocytes. So when you have loss of enterocytes, so if it was this becomes this now. Okay, if this thing this thing is normal, right? If this thing becomes this, you have villus atrophy, okay, in your intestine. You don't have villi, obviously, because you don't have those enterocytes on which you have villi. So you have villus atrophy. This is the characteristic feature of celiac disease. Don't forget this, okay? So this is very important. So you will have villus atrophy. When you have villus atrophy, you have loss of absorptive surface, and the patient will have malnutrition. Okay, and if the patient has malnutrition and there's iron deficiency, the patient will have microcytic anemia. If there is deficiency of vitamin B12, the patient will have macrocytic anemia, pancytopenia, and hypersegmented neutrophils. There will be paresthesia, there will be loss of vibration, okay? And if the patient has loss of folic acid, there will be again macrocytic anemia. If the patient has vitamin A deficiency due to malabsorption, there will be keratic conjunctivitis and Bitot spots on the conjunctiva. And so if the patient has vitamin K deficiency, there will be bleeding, and if the patient has vitamin C deficiency, there will be scurvy. So anything that can be deficient, okay, due to celiac disease, anything can be deficient, and you have to remember all of the symptoms that the patient can have due to deficiency of any kind of thing. Okay, so this is the whole pathophysiology of celiac disease. I hope this is clear. If you have any kind of questions, feel free to ask me in the comment section, and best of luck.